Welcome and thank you for joining today's session. In this presentation, we will explore the critical role of the ECG in the safety monitoring process during clinical drug development. We will place a particular focus on the International Conference on Harmonizations, the ICH E14 guideline, which provides regulatory guidance on evaluating the potential for drug-induced QT interval prolongation and proarrhythmic risk. I'd like to start with a first point which seems to me very important. We have not to forget when we monitor development of a new drug, that the ECG findings can reveal off-target cardiac effects early in drugs development. And we will look for bradycardia or tachycardia, supra or ventricular arrhythmias, conduction abnormalities, that means AV block, bundled branch block on drug, etc., ischemic changes. And that's the first step, of course, before going to the QT interval, which is for today the cornerstone of the drug safety. We have to come back to the cell for a short moment in order to understand exactly what is the QT and what means the new modification of the E14. So we go back to the cell and to the ion channel first. What is the cell? The cell membrane of a cardiac cell, which is a cardiomyocyte, is a membrane which is done of bilayer phospholipid, as you can see there with my arrow there, embedded with proteins. And this membrane contains ion channels, pumps, and receptors specialized for the cardiac function. If we go to the ion channels on the right side of the slide, you can see that these are macromolecule proteins. They are highly selective, for example, to sodium, to calcium, to potassium. And this is a reference to the ERG channel. We will talk about it later on. This is a human heterogogo-related gene which codes for the potassium ion. These macromolecule proteins are highly selective again. They functions as gates that allow ions to move in and out the cells, and they may respond to various parameters through conformational changes. For example, the time, the voltage, and the conformational changes are the different state, open, closed, or inactivated. And again, back to the cell, now going to the action potential. What is the action potential? When you stimulate a cell, there is a depolarization of the cell. The potential was negative and comes to zero and comes to positive. This is the upstroke of the action potential. And this change in the potential, the depolarization phase, is due to the entrance, very quick entrance of the sodium through the membrane from the outside to inside, and a slower entrance of calcium to the outside, to the inside of the cell, going burst to the first phase of the potential, action potential, which is the overstroke, which is the depolarization. Then the cell repolarize. This is a little bit delay because the calcium still enters the cell, and then it goes to the phase 3, which is for us the most important today, which is related to the entrance, to the exit of the potassium from the cell. This is an outward current, and this outward current leads to the repolarization. There is a close relationship between, of course, the action potential of the cardiomyocytes along the atrium and the ventricle. And if we consider the ventricle, the electrogram of the ventricle, the QRS electrogram, which is the depolarization of the ventricle, you can see there is a close relationship between the overstroke, between the depolarization recorded in the action potential and the depolarization of the ventricle. And then if you consider the repolarization, which is the outward current of the potassium, it is closely related to the QT interval, which represents the repolarizations of the electrogram. We talk about the EART blockage, which is the blockage of the voltage-gated potassium channel, and it has an impact on the ECG, of course. That's the reason why it's schematically drawn here. A potassium ion go outside the cell, that's the repolarization, and if you block the channel, there is not anymore outward current going from inside to the outside of the cell, and of course it delays or blocks the repolarization. And the regular QT is the following on the schematic drawing. If you block the channel, you can see that the QT interval is increasing. And it is recorded on this particular experimental data. This is a real action potential recorded in a cell, in a cardiomyocyte, on control and on DL-sotalol. DL-sotalol is a class 3 drug which has an impact on the QT interval, which increased the QT interval. As you can see, the ECG on control and on drug shows an increase of the QT interval, and this is in close relationship to what happens on the action potential, which increase a lot, and the polarization is delayed due to the blockage of the voltage-gated potassium channel. So increase of the action potential, increase of the QT interval. And this is a recording in a patient on a drug who was inducing a QT prolongation. You see the baseline, the QT increase a little bit. Then, as you can see, it increase a little bit more. If you look at this arrow, and give birth to some PVCs, and increase more and more. As you can see, the QT is a very delayed QT, very long prolongation, giving birth to extrasystoles, and then a run of torsade de pointe, and then again modification of the QT, which is increase in delay and which is also increase in amplitude. And this gives rise to what we call torsade de pointe. That means a particular ventricular arrhythmias, and the name come from a French cardiologist in 1966. We described this particular form of ventricular arrhythmias, and the name torsade de pointe comes from the fact that the QRS twists around the baseline. You know, the QRS is negative, then it twists a little bit and it goes to positive. And with the QT increase and this kind of tachycardias, you can, of course, diagnose the torsade de pointe, which is different from other ventricular tachycardias. Why and how to measure the QT interval evolution? Why? This is an important point. The reason is because the QT interval is a surrogate to torsade de pointe. We cannot wait months and months to observe torsade de pointe, which are very rare. And as it's closely related to the QT interval evolution, the QT interval has been decided to be a surrogate to torsade de pointe after works from North American and French experts long time ago. So how to measure the QT interval? There are different ways to do it. I just focus on one, which is the more widely utilized method and the oldest also. And this is the method that is used presently by Banook experts. And as you can see, this is the tangent and intersection with baseline method. You draw a tangent to the descending aspect of the T wave, and you look at the crossing with the baseline, and you measure from the beginning of the QRS to the end of the QT. On this particular schematic drawings, it's around 400 milliseconds. A last point on the QT, you will see the QTc described, not only the QT, but the QTc. The QT interval increase with the bradycardia and decrease with the tachycardia. And to correct the QT, you may use the Bazet formula, which divides the QT interval by the square root of the RR. That means QT Bazet. QT interval square root of the R, the Fredericia formula, and we use more and more often this one, because the first one overcorrect at high rates and undercorrect at low rates, sorry. And the Fredericia formula, which divides the QT interval by the cube root of the RR interval, may provide more accurate correction across a wider range of heart rates compared to the first one, that means the Bassett form. Focus now on the International Conference on Harmonization, the ICWAS E14. And I think that it was important to remind what we have seen from the very beginning of this presentation, the going back to the cell and the history of the QT interval and the torsade de pointe. Well, the ICWAS E14 requirement for registration of pharmacological for human use was described before 2015, actually in 2005. And it was based on thorough QT study, the C-QT study, which was dedicated study to a drug effect on the QT. You had to measure the QT interval evolution during the drug exposure of candidate drug in healthy volunteers. And you have not only to do that, but also to have a placebo group and a positive control group. Usually we use the moxifloxacin, which is a drug which increase and delay the repolarization and increase the QT. You have to do that to establish the assay sensitivity. And you have to perform an analysis by time point. And if there was no significant effect of the candidate drug, that means that the upper bound of the one-sided, the 95% confidence interval of the mean QTc prolongation was inferior or less than 10 milliseconds at any point, the study was considered as negative, and in this case, no specific evaluation was needed in the subsequent clinical program with respect to the QT-QTc interval and the proarrhythmic potential. Again, placebo group, positive control group, and more than that, therapeutic dose and supratherapeutic dose has to be studied as a worst case scenario. Last but not the least, there were some secondary endpoints. To be negative, you have to show that no values were exceeding 500 milliseconds for the QTc, or change from baseline exceeding 60 milliseconds in the active treated groups. And again, and more than that, concentration effect, the PK/PD relationship, had to show the expected positive slope with the control, actually the moxifloxacin, again with the drug, which increased the QT. This is an example of a double delta QTc measurement for the levocetirizine 5 milligram, levocetirizine 30 milligram, and moxifloxacin 400 milligram. Again, the control drug, which was a drug which increased the QT. And you can nicely see that the upper bound of the two-side 90% confidence interval of the predicted mean effects on the delta QTcf for levocetirizine in, let's say, square on circle, white square and white circle, they were smaller than 10 milliseconds, and the upper bound never crossed the 10 millisecond limit. If you look at the control, which is the moxifloxacin, on the other hand, it was crossing the 10 milliseconds. It was greater than 10 milliseconds at the geometric mean Cmax, demonstrating a QT effect. So this has been updated in 2015. Early phase QT assessment had to allow the use of the concentration QT modeling, the C-QT, in early phase trial to assess the risk of a drug to increase the QT. And if a drug was showing low risk in early studies, a TQT, dedicated TQT study may not be necessary. This is the main message of the 2015 update. No TQT study if a drug shows low risk in early study. Of course, you had to encourage the use of continuous ECG monitoring and the model-based approach for better risk assessment. And doing so, this was endorsed by the regulatory agency like FDA in US, EMA in Europe, and PMDA in Japan. We have to focus on the fact that data must be analyzed by an experienced ECG core lab, which accounts for covariates, age, sex, heart rate, and we have to evaluate how robust the model is to different assumptions. How does the C-QT modeling work? Data collection has been performed in early phase trial, phase 1 or 2. ECG are recorded at multiple time points in healthy volunteers or in patients. Blood samples are taken to measure plasma concentration, and capture peak drug exposure and time of maximum concentration have to be obtained. You have to analyze the QTc and the drug exposure relationship using statistical models, which are used to assess the QT changes with increased drug levels and to control for confounding factors, for example, the circadian QT variation. And again, if the upper bound of the 90% interval for the QC prolongation at expected therapeutic concentration was less than 10 milliseconds, then you may consider that the drug had a low risk and no further evaluation was needed. This is an example of concentration effect relationship of drugs with and without QT effects. Look at the first table and the levocetirizine recording, the concentration effect modeling. If you look at the table, at the figure, the slope is not significantly different from 0, and the confidence interval includes 0 with the negative drug. And you can see that never the confidence interval, the upper limit, crossed the 10 milliseconds, and the slope is almost flat. If you look at the dofetalide, which is a class 3 antiarrhythmic drug, which delayed and prolongs the QT interval, if you look at this concentration effect relationship, you see very easily that the slope is significantly different from 0 with the positive drug, with this positive drug, and the confidence interval crosses the 10 milliseconds, confirming that the CR modeling was a CR modeling of a drug which increased the QT. What's the difference between the C-QT and the T-QT? Very briefly, the C-QT modeling may be used in early phase trial as the traditional T-QT was performed in dedicated phase trial. The sample size were about 30-50 subjects compared to 60-70 subjects. And of course, the efficiency was faster and cost-effective when you use the C-QT modeling compared to the traditional T-QT study, which is time-consuming and expensive, and the regulatory benefit is evident. C-QT modeling can replace the T-QT if the risk is low, and T-QT is required if the QT risk is unclear. How the regulators determine if a T-QT study is needed? Again, if a C-QT modeling shows a QTc prolongation of less than 10 milliseconds, no T-QT is required. If the C-QT modeling shows a QTc prolongation of more than 10 but less than 20 milliseconds, more ECG analysis and a small confirmatory study may be needed. And of course, if you have a C-QT modeling which shows a QT prolongation of more than 20 milliseconds, a full T-QT study may be required. Last update, it's the year 2022. And for the first time, it connected the E14, which is a clinical approach, to the S7B, which is a preclinical approach. And in this guidance, there is a connection between the ICH S7B and the E14 guidelines for scenarios where the non-clinical data are informative in clinical study implementation and evaluation. And the reason why I get back to the channel and to the cell in the very beginning of this presentation is the fact that now we include some preclinical data. We have to just remind at this time what is the ICH S7B. The S7B provides guidance for non-clinical evaluation of a drug's potential to prolong the QT interval. And this is based on in vitro testing on the drug effects on the ER potassium. Remember, the ER potassium was the ion channel, the ER channel, the ER potassium channel, which was, let's say, the channel who gives the possibility to the potassium to go outward from inside to outside the cell for the repolarization. And the in vitro test, the ER potential and the potential for a drug to block or not to block the ER channel. And in vivo study measure in the conscious animal the QT, and they may look at the increase of the QT in conscious animals, usually non-rodent animals. And more than that, they may induce the potential for QT prolongation and also the proarrhythmic potential of the cardiomyocyte by different proarrhythmic models. And back to these preclinical studies in connection with the E14, if the both studies, the preclinical and the clinical studies are negative, the previous requirement that with the fact that we have to deal with concentration twofold above what can be seen in patient, the previous requirement can be reduced to covering the concentration seen in patients. There is a summary of the 5.1 and 6.1 questions, question and answer of the S7B and E14 documents. This is a table which is a summary of the two questions, 5.1 and 6.1 questions, answer and answers. This was done by Darpo and it was lastly summarized. I can focus on the first, which is the regulatory pathway question 5.1, when a drug can be safely given to healthy studies at sufficiently high doses concentration. The new recommendation, and I may focus on that, the new recommendation, the TQT study can be waived based on a negative study in healthy subject if this is achieved, if it's achieved concentrations which cover the mean Cmax in patients with high clinical exposure, if supported by negative non-clinical assays, which are performed in accordance with the best practice. That means what we have seen, the ERG and in vivo study covering free Cmax in patients with high clinical exposure. A word on oncology drugs. They have specific characterization and characteristics that may summarize as shown on this slide very briefly. First of all, the modeling of concentration response relationship should be conducted to assess how drug concentration over time may affect QTc prolongation, long-term exposure. Second, oncology drugs which prolong half-life and complex pharmacodynamics may necessitate extending ECG monitorings. So special considerations may be done for large molecules and monoclonal antibodies, as these drugs have unique pharmacokinetics which require specific QT evaluation strategy. There are special populations which are very important to consider, the pediatrics and the elderly, as far as the risk assessment should be made based on age-related factors and modification of the pharmacokinetics and the profile of this oncology drug in this population. And more than that, the cancer patient often have baseline QT prolongation due to the disease by itself, to concomitant medication, antiemetics, endocrine, etc. And the drug-drug interaction, we may interact on the QT, electrolyte imbalance, etc., etc. That's the reason why there is a second part of the table that Darpo, in which Darpo summarized the 6.1 question, the 6.1, when the drug cannot be safely given to a healthy subject at sufficiently high doses concentration. That's mostly oncology drugs that we have seen the characteristic. And the new guidelines, if we focus on it, the new baseline is based on a negative clinical assays performed in accordance with best practice and a negative study in patients, QTc effect superior at 10 milliseconds excluded, and cardiovascular safety database not suggesting an increased rate of adverse events. At this time, if we consider all these negative points, the claim can be made that the drug can be considered to have a low likelihood of pro-arrhythmic effect. Just before finishing, a word on nucleotide, because there is a new publication in 2023 and 2024, and there is based on 29 ONT studies which has been collected. These 29 ONTs didn't show any QTc interval change in humans, no ERG inhibition in vitro, and no QT prolongation and arrhythmogenicity in animal models. This makes the oligonucleotides a low-risk drug class from a cardiac safety. And in place of the TQT study, ONTs may undergo a C-QT analysis using early phase ECG data. If the non-clinical studies are negative and early clinical are adequate, and ECG may be of high quality and the time match with pharmacokinetic sampling, TQT study are not required. So in summary and to conclude, ICH14 is very important to identify the cardiac risk early in the drug development, to protect patients and facilitate the safe development of new therapies, help pharmaceutical companies to design safer drugs, prevents market withdrawals to enforcing cardiac risk, of course, reduce the unnecessary clinical trials and allows early phase data to guide decisions, and promotes international harmonization and reduce regulatory uncertainty. Thank you for your attention.